I'm Markus Rapedius. I'm working as a senior scientist here at Nanion in the SyncoPatch team. And today I would like to talk about some channels, of course not all channels that we have measured on the SyncoPatch, and today it will be dealing with channels involved in pain pathways as entitled in my title, basically. So I will start with a brief introduction on sodium channel family and start with NAF 1.9, biophysical properties and pharmacological properties, and then head over to NAF 1.8 channels, where I'd like to try to give you a little bit of more screening kind of data set. And last but not least, I will go to some slides about NAF 1.7 and show you some state dependence and frequency independence, and I will finish my talk with a brief summary. So basically, sodium channels in general are very important for neuronal excitability, as they are the main drivers for action potential generation. And the family that we know contains 9 members, NAF 1.1 to NAF 1.9, and all the channels are encoded by 9 genes. And the channels are very widely expressed in neuronal as well as muscle cells throughout the body. And I would like to focus on the channels that are expressed in the peripheral nervous system, whereas the talk from Elisa will more focus on the channel that is expressed in the brain, in the central nervous system. I guess the 3 channels I'm talking about have been shown in a lot of studies that dysfunction of a lot of these, basically of all sodium channels, are involved in different disease states, but for the NAF 1.7, NAF 1.8, mutations have been shown to either increase or actually completely abolish pain sensation. There have been a lot of publications on that. So I would like to focus on those guys today. So besides the sequence homology, sodium channels can also be kind of characterized in their sensitivity against toxins. And you can see here current traces from the 3 different sodium channels I talk about today. So it's the 1.7 channel, which is TTX sensitive, whereas NAF 1.8 and NAF 1.9 are basically TTX resistant. And besides this currents, TTX sensitivity or non-sensitivity, you can plot these currents in a so-called IV plot, and you can recognize in this graph here that they are very different, with NAF 1.9 being the most hyperpolarized activated channel. And these different properties also are very important for the physiological function of the channels. All 3 of them are expressed in the peripheral nervous system, and to the peripheral nervous system you need dorsal root ganglion cells. They are a switch station for peripheral and central nervous system. So here you have all 3 channels expressed, and they all play different roles here. So it is actually assumed, here is very schematically an action potential of a dorsal root ganglion neuron. It is actually assumed that the persistent current from NAF 1.9 doesn't play a role for the actual action potential, but it is more a player for the resting membrane potential, whereas NAF 1.7, due to its very fast kinetics, is more like influencing the action potential threshold, whereas the very depolarized activated NAF 1.8 channel are actually the predominant current for the upstroke of the action potential. all these three players come together to generate the action potential and actually also shape it. And of course, if one of those players is kind of impaired, which you can imagine could happen during inflammation or some kind of nerve injury, would have strong effects on the generation of the action potential and also for the pain signaling, which is an important property of the DRG neurons. So in case in a disease state, you can also clearly see that if one of the channels is non-functional, it can have effects on pain. This is also then important if those kind of mutations in those channels have an influence on the pain sensitivity. It makes them in the same way also very attractive targets for drug discovery. So this is a kind of short introduction I'd like to give, and I would now like to start with some data. And the first slides I'd like to show are from NAV 1.9 channels, and it's actually slides that have been done together with Icogen. It's one of our customers, and I would like to say thank you to Icogen who were sharing the results with us and also provided us with a cell line. So I guess to make a stable NAV 1.9 cell line is a very tedious and long process. And when you have achieved this, the first thing you want to see is that the biophysical properties are what you expect. So basically, the first thing is you do some kind of biophysical characterization, and that was done here with Icogen using this protocol, where you start from a very negative holding potential, going in short steps towards positive potential, and then have a test pulse here in the end. And here on the right side, you can see an exemplary trace. And you can use this protocol to actually plot the steady-state activation and the steady-state inactivation curves. And you can see here data from the Synco patch was compared to data from another automated patch clamp device, and the Synco patch could clearly confirm the data with the peak current in between 30 and -40 millivolts, and also the steady-state inactivation and activation curve are very similar. As mentioned before, the NAV 1.9 channels do not show any sensitivity against tetrodotoxin, in contrast to NAV 1.7 channels. So in order to validate this, the first thing is that we tried is to activate the channel in the presence of tetrodotoxin. So you can see this here as we did on the Synco patch, and here's already mentioned. What you can see here is our acquisition software. You can see here the 384 well plate format, basically with 384 individual recordings, individually clamped, starting from A1 down to P24. The color coding, you can always see here some gray, some blue, and some green. The color coding actually reflects the sealing properties, and you can see here you have 90.6% above 100 megaohms on the 4-hole chip. Excuse me. The blue color basically shows a Z resistance that's in between the sliders here. On the right side, you can see here the highlights, 16 wells that you can basically click on with the black rectangles here, and one well here on the bottom is highlighted more closely, and you can also recognize here 2 red lines. They are very important for the analysis. These lines are called our cursors, and they basically represent the region of interest that you like to analyze later. The currents in this experiment were elicited from a jump from -120 to -30 for 40 milliseconds. And when you look at the cursor range in the depolarization step, you can look at the current here. And what you can do in the online analysis view of our software, you can plot this peak current over time, and then you get a so-called IT plot. And here you can see that while recording, we have added several solutions to the cells, to each individual cells with a 384 pipetting head, with pipette 384 wells, sorry. And in each well, we applied a reference solution, 1 micromolar TTX and 1 millimolar lidocaine at the end to block the current. And you can see here it looks nice and homogeneous, and TTX doesn't have any effect, whereas lidocaine blocks the current. In a similar way, we could also determine sensitivity against lidocaine and tetracaine, which basically represent standard compounds for this channel. We observed 350 micromolar for lidocaine and 12.5 micromolar for tetracaine, which is in good agreement with the literature. With this, I would like to sum up with the NAV 1.9 data, highlighting that we could actually generate a stable assay for the difficult target NAV 1.9, and it has been established at Icogen. The biophysical and pharmacological properties are in good agreement with literature, and the assay as it is suitable for any HTS campaign. And I would like to again thank Icogen for sharing the results and also to provide the cell line with us. Now I would like to switch to NAV 1.8 channels, which actually also represents a very difficult target. And again, first of all, I would like to say thank you to Evotec as also one of our customers, who is always sharing results with us, and that was also the case for this assay that we kind of developed together. In this case, the cell line came from Charles River, who gave us a cell line. It's a beta-3 sodium channel co-expression system in CHO cells. Problem with this assay in the first place was not like shown here in the trace view here, that the expression was very low. So what you can see here is basically a recording from a multi-hole chip where we activated the channels from a holding potential of -10 and depolarizing steps to +60. And you can see here the response of the channel towards this depolarization. But in the first place, we started out with a single hole chip, and we observed that only a very small fraction of the current are actually in a window where you would say this is good enough for screening of compounds. So what we did in this case, we actually provided a new type of chip for Evotec, and with this 9-hole chip, basically, we could boost the currents towards levels where we kind of had -1 to -3 nanoampere, and that is good enough to test the compounds. However, before you can proceed to test compounds, we needed to prove that the current voltage characteristics are conserved in the multi-hole chips. And as I've shown here, we analyzed the 1X chips and the 9X chips, and we basically were in good agreement with the literature. But of course, you can see that the success rate dramatically increased when we go to higher current levels. So that was really nice. Given its very depolarized voltage dependence, it is important for this channel because it actually can have effects on the action potential propagation at depolarized potentials. And these depolarized potentials, they can very well occur when you have an inflammation or any kind of nerve injury. And therefore, in these depolarized states, the channels become inactivated. And therefore, it is also very important to analyze inactivated states, because they could be relevant in a disease state. So together with Evotec, we designed a protocol where we could access not only the rest activation from the resting state, but also activation from an inactivated state. So basically, we achieved this by trying several things, and in the end, we found that this kind of protocol worked well with a 5.5 second depolarization to -30, and then again an activation to +10 millivolt. And when we apply this protocol across the ship, we can see that there are two nice peaks detectable to compare directly the activation from the resting state as well as activation from the inactivated state. And as I mentioned, we kind of with this assay looked more into compound plates and robustness of the assay. So here is how we designed the compound plate. It's a 384 well compound plate, where we put 6 10-point tetracane dose response curves and also some single points to have 82 wells with different concentrations in order to determine the IC50 from resting and inactivated state. And basically the remaining wells were filled with controls only to monitor current stability over time, and in a compound screen, basically those control wells would be replaced with compounds. So basically this is how it looked. This is again showing the IT plot with currents from the resting state here in dark blue over time, and also for this for the inactivated state in light blue. And you can see here the reaction of the cells for the 10-point dose response curves. And I've also highlighted here on the right side some examples where you can already see on the first glance that the two states show different sensitivity against tetracane. And this is also highlighted when we use our analyzer software. You can see here the IC50 Hills fit, basically for blocking of the resting state activation versus activation from the slow inactivated state. There's also a slight shift in the Hill coefficient and a shift in potency of about 15. Besides this, we also plotted here the variation of the control valves from the inactivated states for a compound period actually of 10 minutes. So for this assay, it was required to incubate the compound for 10 minutes. So I guess this is very nice that the run-up, run-down is in kind of small limits. And here we repeated, as I mentioned, we repeated this experiment for several plates, and we were pleased to see that the IC50s were very stable among several plates. And we could also, from these compound plates, plot set prime values, where we compared the positive and negative controls. And we were happy to see that the set prime values are also very stable and above a value of 0.5, which gives a good idea how good the assay is. And we were also very happy to see that at the same time, having a high quality assay, also the success rates were routinely above 90%. So with this, I'd like to sum up my slides on the NAV 1.8 data. Again, we were very happy to help out and generate a stable assay on the difficult target NAV 1.9. It has been established at Evotec, and the screen has been run. We also designed a protocol for recording activation from resting and inactivated states in one protocol. And here I also showed a kind of small robustness a test where we have compared several compound plates that indicates that the assay is suitable for an HDS campaign. Okay. Again. So finally, I would like to go back to the fastest one, NAF 1.7 channels, rapidly activating, inactivating, but also do recover very slowly from inactivation. And I would like to show you that for this channel, we have developed a 3-state protocol where we actually could compare activation from resting and from 2 inactivated states. And in this case, the cells were kindly provided by Nanion. I'd like to emphasize this. And when you compare the results here as shown by a snapshot from our data control software, you can see here in the bottom the trace view of an individual cell with 3 different activations. And in the middle, you can see the trace view for the 3 different pulses over time, the IT plot. And on the top, you can see 3 different dose response curves from one cell as we have recorded 3 different peaks, basically. So in this case, we have done a cumulative assay where we applied different concentrations on the same cell. And here on the right side, just some more examples of the same recording, basically. Coming to the end, I'd like to show this experiment we did. Besides the state dependence, there's also a term called frequency dependent that basically describes that the frequency of opening of channels can have also an effect on compound efficacy, basically. And this property is usually assessed by a pulse train. And you can see here the protocol where we have a train of 10 pulses within 1 second, indicating it's a 10 Hertz pulse. So basically, you would have 10 channel openings in 1 second. On the right side, you can see that in this recording, we have applied the pulse train and also used different tetracane controls as a model substance. And you can see that the train is basically very stable in the control base, whereas at different tetracane concentrations, you can see that in the subsequent pulses, the peak amplitude decreases to a steady state level, and this is concentration dependent. This is classically what is described as a frequency dependent block, because the unblocking basically is too slow for the channel, and basically the compound will accumulate in the channel and thereby blocking it. So and then we have basically the possibility to have 3 readouts from 3 different pulses. And I would like to again show you a snapshot of data control where we have basically set 3 cursors in our control in our protocol from the first, the second, and the 10th pulse of the train. And then you can compare 3 different IC50 values from 1 cell for the 3 different pulses. And the dose response curve here on the bottom should highlight you that tetracane is the compound that clearly shows a frequency dependence on NAF 1.7 channels. And with this, I'd like to close and thank again everybody for listening. And I think my main summary is that the data I presented here clearly indicates that for not only NAF 1.7, but this is what NAF 1.8 and NAF 1.9. The Synchro Patch platform is a very versatile tool to generate assays suitable for drug discovery.